My name's Avi Chakrabartty. I'm from the University of Toronto, and I'm going to talk to you today about making the drugs that treat ATTR. Okay, so these are my disclosures, and let's get started. So here's the general procedure by which drugs are made today. It starts with this basic science phase up in the left, top left over there. And this basic science phase, we're trying to unravel the disease mechanism. And so what we want to do is to learn as much about the disease as possible and get our knowledge to the point where we can make or set up intelligent hypotheses of how to treat the disease. So how long does this basic science phase take? So for COVID, it took less than a year. For ATTR, I estimated it took about 50 years. So it took 50 years of knowledge gained from treating patients and basic science, et cetera, to come up with these hypotheses. And then the hypotheses are tested in the preclinical phase. So in the preclinical phase, most of which is done in the laboratory, you do a lot of experiments. You do experiments with test tubes. You do experiments in live cells. And you do experiments in animals as well. And what you're trying to do is just to gather as much knowledge as you can to either support your hypothesis or preferably to support your hypothesis. Because if it doesn't support your hypothesis, then you have the wrong hypothesis. Okay, so this clinical phase, you're just obtaining enough experimental data to show that your drug is doing what you think, what you want it to do. Okay, so if it passes through that, then if you do think that you have a reasonable drug candidate, you can apply for a phase 1 clinical trial, which is mostly on setting the safety profile. And then if it passes the safety profile, to the phase 2 clinical trial, where you look at the drug's efficacy. This is sort of a medium scale trial. And you can sort of think of it as a dress rehearsal for the phase 3 trial, which is the actual workhorse. So the phase 3 trial, which is a large scale trial, and it's the most robust test of the drug's benefits and risks. And so if your drug candidate passes through the phase 3 trial, then you can submit a new drug application, and your drug will be approved for marketing. But it won't appear on the market right away. There's this other reimbursement phase where you set the price for the drug, and insurance companies decide how they're going to handle it. Okay, so this is just the general procedure. So let's talk about how this all worked out for ATTR. So we'll start with the basic science phase of ATTR. So like so many things, the basic science phase of ATTR started with Elvis. So it was in 1957 that TTR was discovered to be a vitamin A transport protein. 1958, TTR was discovered to be a thyroxine transport protein. And then in the early 60s, they found that TTR was a lot like the Beatles. You know, they're both tetramers made out of 4 monomers. And then by the mid-60s, early 70s, we start to know more about the diseases themselves. We saw amyloid deposition occurs not only in polyneuropathy, but cardiomyopathy. And there were some hints that at least in some cases, the amyloid was TTR. And then in 1977, there's this really nice paper where they made antibodies to the amyloid and showed that that reacted to TTR. And then they made antibodies to TTR, and that reacted with the amyloid. So basically, it came up with this idea that there's these two different states for this TTR molecule, sort of a native, natural state where it's transporting vitamins and thyroid hormone, and there's this sort of disease state, which is the amyloid. So what causes this conversion? So the answer was actually quite simple. So if you expose the TTR tetramer to a little bit too much acid, it falls apart into monomers, and then the monomers assemble into amyloid. And then by the early 80s, the mid 80s, early 90s, it was determined that there's actually that the mutations actually impact this actual pathway, and that mutations actually enhance this process. But by the year 2000, we knew this mechanism for TTR quite well. I mean, I would say at sort of almost atomic resolution. So that's a good thing, because the year 2000 is the year where my knowledge of rock music runs out. Okay, so let's talk about the mechanism. Okay, so it all starts with this molecule right here, this transthyretin tetramer. So I'm showing it here as an all-atom model. So with this model, every atom is represented by a sphere. So carbon atoms are gray spheres, oxygen atoms are red spheres, nitrogen atoms are blue spheres, and you can see two yellow sulfur atoms poking through as well. So this sort of representation, the all-atom representation, gives you an appreciation for the overall shape of the protein. And this sort of representation of TTR, it always reminds me of a roast chicken. You know, you can see the two drumsticks on the bottom, and then there's the wings wrapped around the breast, and then there's the neck on top, right? So this is the roast chicken model. Okay, so if we were to zoom in right to the core of this molecule, what we find is it's made up of a long chain, and this is called the main chain. And so the main chain is the skeleton of your molecule, and I've shown it here in green. So the main chain defines the structure. And then sprouting from the main chain are these small side chains, which I've shown in yellow. So here what I've done is I've taken that transthyretin molecule, and I've stripped away all of the side chains. And so what I'm showing is just the main chains. So this is the skeleton of TTR. And so what you can see is it's made of some sort of repeating structures. There's these strands. You can see a couple of strands, are up at an angle like this, and then there's some strands that are sort of horizontal, other strands that are connected by loops around there. So actually, all protein structures are made up of three substructures. The strand, which I've shown here in green, as these green arrows, the loops, which are yellow, and the helices, which are magenta. Okay, so what we have is we have a computer program where we input the main chain model. And the computer program spits out this ribbon model. So this is completely scientific. It's not artistic at all. And so, and this ribbon model is really useful for talking about disease mechanisms, which I'm going to talk about now. Okay, so yeah, I had to give you a little tutorial on protein structures. This is the final product of the basic science phase of ATTR. It all starts with that molecule on the right, on your left. And that's the TTR tetramer. And so it's made up of the 4 monomers there, you know, John, Paul, Ringo, and George. And then if the tetramer is exposed to too much acid, it falls apart. And the monomers each go their separate ways. And the monomers, when they're by themselves, when they're isolated, they're not very stable. And they begin to sort of flop around. And so while they're like flopping around, every now and then they'll form like a pancake. And so when this pancake forms, and if it finds another pancake, the 2 pancakes stick together. And then every time a pancake forms, it sticks on top. And what this causes is you get this growth, this stacking of these pancakes. And these pancakes can stack up to 200 high. And that's the amyloid fibril. So that's that big, long stack of pancakes. That's what deposits in your heart muscle, making it stiff. That's what deposits around your nerves, causing the neuropathy. Okay, so that's the mechanism. And so according to our scheme, when we know enough about the mechanism, we should be able to have intelligent hypotheses for how to treat the disease. So actually, there's 3 good hypotheses that have been put forward for ATTR that have stemmed from this. And let's look at the first one. So the first one is the silencer hypothesis, right? So this is CRISPR-Cas9 technology, and the hypothesis, and silencer technology as well. So the hypothesis is that TTR is a non-essential protein. We don't need it. We can actually do without it. So let's stop making TTR. And if we just stop making TTR, then there won't be any TTR to turn into amyloid. So that's the hypothesis. And fortunately, there are established ways of reducing protein production. You can target the DNA or you can target the RNA. And when you target the DNA, that's the CRISPR-Cas9 technology. So, so there's this reagent, this NTLA 2001. That's a gene transfer reagent, which was tested in the phase 1 clinical trial in both polyneuropathy and cardiomyopathy patients. And the results were very promising. And in fact, they've decided to skip the phase 2 trial and go directly to the phase 3 trial, which is in the works right now. Yeah, so I'm finding out that more often, if your phase 1 looks quite good, it seems that you can actually skip the phase 2 trial. Okay, now with the RNA targeting, we're much further ahead with RNA targeting. We actually have drugs which have been approved. And what I've shown here are 2 RNA targeting drugs. These are the first 2 RNA targeting drugs that have been approved. And because they have been approved, what I've done is I've gone through the phase 3 trial, and I just sort of pulled out sort of summary statements from the trial, just to give you a gist of what the results of the trial were. And so I did that for both patisiran and inotersen. And you can see that so a lot of positive comments and things like improved functional capacity, improved quality of life, slowing of neuropathy progression. And so There are all of these sort of qualitative statements, but they're all backed up by a lot of laboratory results, results from questionnaires. So it does appear qualitative, but there's a lot of quantitation there. So I just want to give you sort of a glimpse of what a successful phase 3 trial looks like. So the other hypothesis, the second hypothesis, is the stabilizer hypothesis, which, so all of us know right now that TTR is a tetramer that must fall apart into monomers to form amyloid, right? So, so this hypothesis is, so let's just stabilize the tetramer. And this will prevent it. And if we stabilize it, it'll prevent it from falling apart. So how do you stabilize it? So one way to do it is with this drug called tafamidis. It's a small molecule. And you can see the tafamidis, one molecule of tafamidis slips in on the left and another molecule on the right. And it slips in just where all the monomers are touching each other. And so what tafamidis is it's like a super glue. And so you put a dab of it on the left and a dab on the right, and that cements all 4 monomers together. And then you get a hyperstable tetramer. And if you have a hyperstable tetramer, it's not going to fall apart into monomers, and the monomers won't go on to form amyloid. So that's the idea. And so did it work? Yes, it did work. And tafamidis was the first disease-specific therapy that was approved for treatment of ATTR. And these are some of the summary statements from that phase 3 trial. So I think, so you can read that. So it sort of supports tafamidis as a long-term treatment for ATTR. Okay. So there's another hypothesis. It's the depleter hypothesis. So this depleter hypothesis proposes that antibodies that bind the pathological forms of TTR, which are like the monomers and the fibrils, but leave the tetramers alone, then these antibodies can be used to clear all of these amyloid deposits. And so how do we do that? Well, we can do we can do that using antibodies. So I've shown the structure of the IgG antibody on the bottom on the left, on the bottom left. And you can see the strands, and you can see the loops all connecting. So that's a ribbon diagram of an IgG antibody. So what do antibodies do? They bind onto things. So if a bacteria enters your bloodstream, an antibody recognizes the bacteria and binds onto it. And then there are blood cells in your body called macrophages. And these macrophages, they recognize anything that's been tagged with an antibody. So the macrophage will identify the bacteria which has been tagged with an antibody. Then the macrophage goes over and engulfs that bacteria and then digests it. Okay, so how can we apply that to this situation here? So if we could make an antibody that recognizes only the pathological forms of the protein, so the ones which are in the red box. So if we can get the antibody to recognize those, then the antibodies will tag these proteins, will tag the monomers, tag the fibrils, and then the macrophages will detect the tagged molecules and then clear them, digest them. So, but the issue is we have to make sure that those antibodies don't bind the native tetramer, which is up in the green box in the upper right. So how do we do that? So this is work that came out of my lab here, and it was helped along. So all of the clinical work was sponsored by Prothena. Many of the Prothena scientists are here, and is currently been taken over by Novo Nordisk. So what did we do? So it was Novo Nordisk who came up with the name, by the way. So what did we do? So we spent a long time just looking at these structures. And then what we wanted to do was to find a piece of the protein that's exposed in the pathological structures, so the three on the right, the folded monomer, misfolded monomer, and the amyloid fiber. So we wanted to find a piece of the protein that's exposed when the protein is in these states. But we also want to make sure that that piece is buried when it's in its normal, native, natural state. And then so we looked at all these structures, and we did find one piece of the protein, and that's that little red strand right there. You can see it in the ribbon diagram of the folded monomer. So that red strand is in the folded monomer in the all-atom model. You can see that the ribbon that strand is nice and exposed. It's also nice and exposed in the misfolded monomer, and in the amyloid fiber, it's like almost hyper-exposed because they all line up along one face of the amyloid fiber. So what we did was we made an antibody to that little red piece. So that little red piece, it's relatively small, so we can just make it chemically. So that's what we did. We just synthesized that red piece chemically, and then we injected that piece into a mouse. And then the mouse made antibodies to that red piece. And we took those mouse antibodies and we converted them to look more human-like. It's a process called humanization. So we humanized these antibodies because you can't put mouse antibodies into a patient. And so you have to humanize it first. So that was done. And so we have these antibodies to these this red bits which are exposed in the pathological forms but buried in the native natural forms. So this is an ongoing story. It's not done yet. I'm just going to show you the data so far. So in the preclinical phase, we saw that the antibody binds monomers and fibrils, but not tetramers. They also recognized amyloid in tissue, and they actually inhibited fibril formation. And more importantly, immune complexes formed between the antibodies and the fibrils were recognized and cleared by macrophages. Okay, so this was enough positive data to justify a phase 1 trial. And the results of the phase 1 trial was that it's safe and well tolerated. But more importantly, there was actual signs of improvement, which doesn't often occur in a phase 1 trial. So there was improvement in neuropathy symptoms, as well as cardiac improvement as well. Okay, so now, Prothena sponsored the phase 1 clinical trial, and now it's the Novo Nordisk is doing the phase 2 clinical trial, and they're analyzing the data, and the results should be out early of 2025. Okay, so there's another antibody, depleter antibody out there as well called Alexin 2220. Now the hypothesis behind this is really fascinating. So the hypothesis posits that some TTR misfolding is occurs naturally. So it's occurring in me right now. Some of it is occurring in you as well. And so the immune system recognizes this. And then the immune system makes antibodies to these misfolded forms. So, and the idea is expanding from that is this age factor. So if an individual, the older an individual is, the greater likelihood that they would have misfolded TTR. So there might be this sort of accumulative effect. Okay, so what this company did, it was Neuroimmune. They went and they got this library of antibodies. And these antibodies are generated from elderly, healthy adults. So the idea is if you're elderly, you would have experienced or been exposed to more misfolded TTR than if you were young. And then so because of that, an elderly person is more likely to have made antibodies to misfolded TTR, and that's why they chose the antibody library from these healthy elderly patients. And so did it work? Well, they did find this one antibody, this Alexin 2220, which binds to fibrils, but doesn't bind to tetramers. That was their screen test. And so here on the right, I've shown the structure of a small fibril, and I've shown where the different antibodies bind. So up at the top, that's where the Coxson antibodies bind, and the Alexin 2220 antibodies, they bind this sort of loop, structure that aligns along this one face of the helix. So that's where I'll show you the data we have on Alexin 2220 so far. So preclinical, very similar to Coxson, binds monomers and fibrils, but not tetramers, and it formed immune complexes with misfolded TTR that were recognized by macrophages, and they actually showed that this could actually occur in the heart. And so they started their phase 1 clinical trial, and the antibody, the Alexin 2220, met all the safety requirements. But what was interesting and significant was that there was a visible reduction in amyloid, detected both by imaging and as well by this MRI, these MRI methods. And so this is really good, significant data for a phase 1 trial. And so they were inspired by this, and so they decided to skip the phase 2 trial, and they're going directly to a phase 3 trial as well. Okay, so just summing up here. So this is this isn't a complete survey of all of the drugs. These are just some of the first one. But I just want you to sort of see how we have drugs which are out on the market, some that are at different phases, and what's fascinating is these drugs are working by 3 different mechanisms, right? And so when you can attack a disease with these 3 different approaches, then you know the idea of combination therapy makes sense there. So that's quite nice. Okay, right. So these are the internet resources I use to make all the pictures, and these are my acknowledgments. And yeah, thank you. Sure. Sure.
Okay, questions for the end. Okay.
Oh, okay. Okay.
Okay. Thank you. Thank you very much, Dr. Chakrabartty. That was very interesting. I've worked in HATTR for 5 years, and I now know what all the molecules look like. So that was very educational, so thank you very much. And good luck with those trials. So I'm going to pick up a little bit from where Dr. Chakrabartty kind of ended off in terms of where are we with the different drugs in terms of their drug development process, and then what happens after we get to phase 3, and how do we actually get drugs to ultimately to Canadian patients. So this is another way of looking at the whole process of drug delivery to patients, and I'm going to sort of focus where that big red circle is in terms of regulatory review, bringing the drug into Canada, and then how do we get market access for patients, which Anne-Marie commented on. What can we do to do that faster and better in Canada? So we'll talk a little bit about that, although there is a lot of process involved. We've already talked about phase 1, 2, and 3 clinical trials. What's really important to know is that 99.9% of the time you do need a successful phase 3 clinical trial in order to go forward into your regulatory review. In oncology or in some extremely rare diseases, you can work off real-world evidence, but we're not going to go into that today. We're going to sort of focus more on the standard process. So this is what the whole process looks like in Canada. So you start with Health Canada, obviously, and Health Canada is who gives you your license in order to be able to import the drug into Canada and ultimately sell it, which is great, but then you need the Canadian Drug Agency and INES to evaluate whether or not there's good clinical value to the drug coming into Canada and treating patients, and then the PCPA, they decide how much we should be paying for that and who should be eligible for the product. And then there's all the different drug plans, both the private and the public. And so that's what I'm going to go into today and sort of walk us through this overall flow. So regulatory new drug submission and review. So if you want to bring a new drug into Canada, you pay Health Canada $520,000. You put together a massive submission, and in the old days it was actually binders, and I remember these in the warehouse, they'd be shipping boxes and boxes of binders because they want to look at everything to do with the drug. And with Dr. Chakrabarty's presentation talking about antibodies and how do those work in the human body, and are there any side effects, and how do you make the antibodies, all this sort of stuff. So they look at all the animal models. They look at the human trials. What's your manufacturing process look like? Do you actually have a good way of making it? What is the efficacy? What is the safety? What kind of warnings and precautions do patients and physicians need to know about if they're going to use this? What kind of patient information is really important? Beyond what this is a great educational day, but how do you learn about the drugs? And then, of course, the chemical data. So a lot of what Dr. Chakrabartty presented, that would be included in your Health Canada submission. There are 2 timelines. There's the standard review, and that's 355 days. So Health Canada, 99.999% of the time, hits that 355 days. There's also a priority review opportunity at Health Canada, but a priority review is only available if there's no alternative therapy available in Canada, okay? So if you already have a medicine that has that same indication, you don't have an option to go with the accelerated review. But when there is no option, you have the opportunity to go forward with the accelerated review. At the end of your 355 days, you can get one of 3 outcomes. The best is if you get your Notice of Compliance, also referred to as your NOC. This basically says you are licensed and approved to be able to bring your medicine into Canada and to sell it to patients. There are 2 other situations that can happen. One is a Notice of Compliance with conditions. This means Health Canada says, yeah, you can bring in your drug, you can treat patients, but we have some questions about it. So you need ongoing additional monitoring in place. It may be a very restricted patient population that is eligible. They just need more information, but they do recognize there's a huge unmet need, so we need to bring the medicine into the country. Or the red X means Notice of Noncompliance. You are not authorized to bring the product. You are not authorized to sell. Back to the drawing board. So those are kind of the 3 outcomes. And when you get your notice of compliance, you get your product monograph. And the website that's at the top of there, I should have created a scan code. If I was more tech savvy, I would have done that. But that's every single product in Canada that has ever been approved. You can find their product monograph there. And your product monograph is basically the Bible or the instruction manual on what your drug is, how it works, who can use it, all the safety information, dosing, how to store it. Does it have to be away from sunlight? Does it have to stay in the fridge? Can it not go in the fridge? Can you eat it with food? Can you not eat it with food? Everything imaginable. What kind of patient populations, geriatrics, pregnant, pediatrics, all sorts of things. So it's all composed in this product monograph, and that is basically what a manufacturer can now go and talk to physicians about, and in what it also contains the information that we're allowed to provide to patients in the patient leaflet who have been prescribed the medicine. So you might think, I've scored a touchdown, this is great, I can bring my medicine into Canada, but the reality is we're only halfway down the field, because you can bring the drug into Canada, but if patients can't get it, you just have a warehouse full of medicine. So the second half of the field is actually much more important, because you need to get access. So, and you also need to bring it into the country. And this is just one of these time things I just want to make a comment around. So you get your notice of compliance, and then you run back to your manufacturing and say, here's our DIN, or our drug identification number. Here's the final labeling. And they have to put that all on the packaging. They have to put stickers on every single vial or every single box, print it, and bring it into Canada. So, and then Canada, you can only keep your drug in what's called, like a licensed importer can only bring it in. It has to live in a very specific place where Health Canada can come and audit and check on it. Every medicine has to go through a QA process to make sure that what you said is in the box is actually in the box. So Health Canada likes it when you do this quickly, because they've gone through the process of actually reviewing your drug. And if you do an accelerated review, you actually have to bring it to market within 60 days. That's a requirement that Health Canada says, and they'll ask you that right up front. You're going for an expedited review or a priority review. Are you going to be able to bring the drug to market in 60 days? Because they're not going to do a quick review for you if you aren't going to go through the effort of getting it to market quickly. So the manufacturer, it comes out of their production facility. Sometimes it's within Canada, sometimes it's overseas. Then it comes into a warehouse, it goes to a wholesaler, Innomar, McKesson, Shoppers Drug Mart, all of these different things. And then it goes out to either the hospitals, if it's a hospital treatment, or to different pharmacies. And that can be a specialty pharmacy, or it can be your regular retail pharmacy that you go to get all of your medicines. So that's kind of the general importing and selling process. So we have our Health Canada approval, we have the drug in the country. Now, what is the process to get actual reimbursement or market access? And this is a real mix of reviews and negotiations and ultimately product listing agreements. So we'll start with the health technology assessment. This is done by the Canadian Drug Agency. It was previously called CAS. They've rebranded, it's now called CDA. They are a little bit different from the FDA. FDA actually reviews drugs, that's Health Canada's job. CDA, this is everything that CDA does, but for the purposes of today's presentation, we're going to talk about reimbursement reviews and what do they do in terms of looking at new medicines coming into Canada and to make sure it's going to give good value to Canadians. So CDA does it for all the provinces except for Quebec. Quebec has their own version of CDA, INES, the Institut National d'Excellence en Santé et Services Sociaux. They do a similar approach, looking at the Quebec population. They look at not does the drug, is it safe? They want to know, is it effective? What does it actually do for the patient? What value does the patient actually get? And is it cost effective? If I treat a patient with this medicine, do I save money elsewhere? Can I prevent hospitalizations? Can I keep people working longer if they take this medicine? So what's the value not just to the patient, but to the healthcare system? So this is what their job is. And they also take into consideration patient and physician perspectives. So when a new medicine comes to Canada, once the manufacturer has submitted to CDA, as patients and physicians, you're able to submit into CDA and give your opinion. I think this drug makes a lot of sense because I go from, infusions to injections, or I go from a handful of pills to 1 pill, or I go from 5 inhaler puffs to 1 inhaler puff every 3 weeks. So you can take that patient perspective, and Anne-Marie has led TAC in terms of providing those patient input into those over the years, because it's very important that the regulators understand the patient perspective. They're not just looking at a clinical trial in isolation. So after they do their review, which takes about 242 days, and CDA sticks to that timeline quite closely, so which is nice to see. They provide recommendations that guide the provinces on whether or not they should be funding this drug. And those recommendations include, should it be funded? Who should be eligible? What does the patient look like who's going to get the most value out of this? What are the reimbursement criteria? We talked yesterday about different questionnaires or different tests that some physicians have to do in BC versus in Quebec. So they outline what are those different eligibility criteria. Who should be able to prescribe? Sometimes anyone can prescribe. Sometimes it's only a very narrow slice of physicians who are real specialists, because they don't want the medicine in the hands of anyone. It's expensive. They want to make sure it's handled properly, and it's only going into the patients who are going to get the most value out of it. And they also make recommendations to the provinces on really how much they should be paying for this drug and how often patients need to be reevaluated to make sure the plans are getting the value for it and make sure the patients are doing well on the drug. So that's the role of CDA and INESS. One comment that's unique about INESS is they actually take into consideration the societal impact of having this drug available. For example, what is the impact to caregivers because they don't have to drive their loved one to the hospital all the time, or patients are able to go back to work, or caregivers are able to go back to work. So they do take that societal impact into consideration as well, which is unique in the province of Quebec. So you've got your Health Canada approval, you've got your CDA positive or INESS recommendation, now you go to the PCPA. The PCPA is the Pan-Canadian Pharmaceutical Alliance. Think of them as Costco. They come to the manufacturers and they negotiate on behalf of all of the provinces on who should be eligible, how much we should pay, and how quickly we can bring the medicine to market. So they do, in the past, this used to be every province would do their own negotiation. It was very time consuming. It took a lot of resources on the manufacturer side. It also took a lot of resources on the provincial side to do all these clinical reviews. So they put this in place about 15 years ago to negotiate on the behalf of all of the publicly funded insurers, so all of the public insurance plans. So they assess is it cost effective? They also know how much they pay for other drugs in similar classes, so they're able to sort of see whether or not they're on par in terms of a benchmark. Improve the consistency of funding decisions, so it's not one drug gets in and one drug gets off and you don't know why. It provides a little bit of transparency. And most importantly, reduce the duplication of efforts, which makes it more cost effective for everyone. At the end of your negotiation with the PCPA, you can get an unrestricted recommendation. Anyone can have this. Think about that for an antibiotic. Restricted to a specific indication or line of therapy. Sometimes it has to be first line, sometimes it has to be second line, or sometimes it's really exceptional access. And when you're working in the rare disease space, all of the medicines come out with an exceptional access, which means it's every drug, every patient request is going to be individually adjudicated, which again leads to some of the time delays that we have for these patients. With the PCPA, there's two components to that. Once you can go into what's called consideration, where they sort of think about, do I want to, basically, am I going to ask them to the dance? I'm going to think about it. Do we want to prioritize this negotiation? Is this important? How many patients are waiting? And that can take 185 days, where as a manufacturer, you're just waiting for the PCPA to say, let's start negotiating. Then you actually start negotiating with the PCPA, and that's usually another 5 months. So that's a big part of the situation in terms of part of the delay that the PCPA can add to the situation. So if you think about a year, and you went to Health Canada in January of 2023, you'd get out kind of of Health Canada beginning of 2024. Your CADETH or your CDA review would take you to the end of August. Then the PCPA would think about it. They'd consider you for 183 days. We're now at the beginning of 2025. Then they negotiate for another 144 days, and you're in the middle of 2025. So that's basically the time it takes. So from Health Canada, going into Health Canada, to the PCPA saying, yes, we think the provinces should list you, that can take 924 days. So just under 3 years. But you're not there yet, because all you have is an agreement that the provinces will list it. The provinces actually haven't listed it yet. And that's what's next. The 14 drug plans have to actually list the product. And this is where some very interesting data has come out on which provinces list first and which provinces list last. And you can see there's over 200-day difference between the one who goes first, which is often Quebec, to the one that goes last, which is often PEI. So, you know, that can add from Health Canada to the very end. For everyone in, you're basically at about 32 months, which is just shy of 3 years. So it's a long time. And in terms of, you know, Anne-Marie asked the question, is there anything you could do to accelerate it? There are a couple things that you can do, and I'll talk about that. But one of the biggest thing is where we have the CADETH review time. You can actually submit to CADETH while your drug is going under review with Health Canada. And that can accelerate things by about 6 months for patients. So that's one of the biggest places where manufacturers can do that. Manufacturers have to have the resources to do that, to be able to have their cost effectiveness model ready to go, be able to submit that, because that's a huge submission. It goes not only to CDA, but to all the provinces at the same time. So it's a big lift for manufacturers, and they just don't necessarily always have the resources to do that. So if we compare that to, so now you actually have scored a touchdown because you've got all your provinces in, your PCPA in, and in terms of private payers, I'll just make a comment that different private payers operate differently. Some wait for a CDA recommendation, and others are willing to go right after you have your Health Canada approval. It all depends on the different private payers, but you go through the same process. They do their own review, and then you end up negotiating with every single private payer as well. So how does this compare to our other countries? Sometimes you want to be at the top, and sometimes you don't. And this is a situation where you don't really want to be at the top because we are the worst. Takes us the longest in Canada. Japan, other than the US, is the fastest, and then you can see all the way up. The average there is 351 days, so a year, but we're well past that. So a quick comment on different types of insurance, because working with our patient support program, we know that this comes up a lot. So there are public insurance, private insurance, and then there's cash paying or out-of-pocket. There's basically two kinds of public insurance in every province. If you're over 65, you automatically go into the public insurance plan. If you're under 65 and you don't have adequate private insurance, you can go on to their private their public plan. Depending on the province, there can be co-pays or not associated with it. Sometimes there's a 3-month delay associated with that. But there are public plans available for patients who don't have adequate private insurance or no private insurance. The private insurance is like saying, I'm going to buy a car, because there's every single kind of private insurance out there. Often they can have up to a 20% copay, and each medicine needs to be negotiated. But when I say there's a huge variability, some patients have an annual maximum of $2,500 and mandatory generic drugs, up to patients who have unlimited coverage, and they can get reimbursement as soon as a drug is approved by Health Canada. So it's a massive spectrum in terms of what private insurance is available for that. So we know that there's a massive delay. We know that public and private insurance doesn't always cover. So what are our options? What other options are there for patients? And I wanted to talk a little bit about manufacturer-supported access, because in rare disease, and even not in rare diseases, the manufacturers actually step up quite often and provide access to patients earlier than the insurance plans do. So there's 4 different kinds. I'm going to start on the left-hand side. So special access programs. These are for medicines that are not approved by Health Canada. Okay, so this is before you have Health Canada approval. If a physician knows that there's a medicine that's under development, it may have gone through phase 2, it may have gone through phase 3, it may be approved in another country or not. Your doctor, or the doctor, can apply directly to Health Canada and say, Health Canada, I have a patient, they're in desperate need of this medicine, I know it is available, I know it's manufactured, can I please bring it into the country for this one patient? And Health Canada may approve it, and they may approve it for a very specific period of time, and the physician actually has to be the importer for that medicine. Okay? So this is one option for patients. The second option is compassionate use. And this is for medicines that are approved by Health Canada already, but there may be a few situations where patients can't access that medicine. For one situation, it could be the patients have no insurance. They have no private insurance, they have no public insurance, they may be a recent person who's moved to Canada, they don't have access to anything. You could apply for compassionate use. If patients don't fit the reimbursement criteria, so we can have very specific reimbursement criteria. If the patient doesn't fit that, for whatever reason, the only option is to go and request from the manufacturer compassionate use. Or the patient wants to use the drug, or the physician wants to use the drug on the patient in an unapproved indication. This is much more common in oncology, when a drug may be approved for one tumor type and they know there's studies going on in another tumor type and physicians want to try that. And in this situation, the HCP would apply directly to the manufacturer to get approval for that. The third type is bridging product. This is probably the most common way that manufacturers provide assistance. So this is for Health Canada approved medicines. You're undergoing your 917-day review process with all the different payers and review processes. But all manufacturers want patients to get access to their medicine. That's everyone's goal. So what they provide is bridging product. So you can have access to the new medicine in advance of it being approved by the public or private health plans. The goal is ultimately that once the public health plan or the private health plan covers your drug, that you would switch to paid product. But it allows the patients to get access much sooner, sometimes 2 years earlier. It's normally managed through a patient support program of some sort. The patients would have to fit probably some sort of manufacturer criteria because they want to make sure ultimately the patient would get reimbursed in the future. The goal is to really help patients because you don't want to bring a drug to market and then no one can get access to it. It helps no one. It doesn't help the manufacturer. It's so frustrating for patients and so frustrating for the physicians. And then the final way that manufacturers can support access is with financial assistance. So many patients, if they have private or public insurance, they have to have a copay or a deductible. And often the manufacturer covers that. And that copay can be, for example, in Alberta, it's $25 a month or $25 per prescription. And the manufacturer could pay for that. Some people have copays that are $100,000 because it's 20% of your $500,000 drug, and the manufacturers cover that as well. There are a couple places in Ontario with the Trillium Drug Plan and in Manitoba as well, where the manufacturers are actually not allowed to pay the copay, which can be very challenging for some patients because that copay can be significant sometimes. Okay, can you press forward? Perfect. Okay, just as, at the very end, I just want to touch very briefly on the National Rare Disease Drug Strategy. This is something that CORD, or the Canadian Organization for Rare Disease, is advocating very strongly on behalf of all rare disease patients. But as a reminder, the Trudeau government promised $1.6 billion to be spent over 3 years to help patients with rare disease. And the idea was to improve access to rare disease, improve screening and diagnostics so we can get earlier access, which was something that, access to diagnosis, something that's so important for all of us. Improve coverage, making sure that there's no postal code lottery that, oh, if you live in one province, you get access, but if you live in another province, you don't get access. And also help with decision-making and pharmaceutical management, sort of kind of more behind the scenes, because dealing with rare diseases is obviously very different than dealing with hypertension or, you know, a bacterial infection kind of thing. To date, only, oh, if you can go back one slide, thank you. To date, only British Columbia has signed one of these agreements. They made this announcement back in the middle of July. We're not sure what's next, who's going to sign up next, but the money is there. Hopefully the provinces will be able to make agreements with the federal government soon to be able to provide this funding to help more patients with rare disease. Now we can go to the next one. And then I wanted to just make one comment, because we hear this a lot from physicians and from patients, is why are some of these drugs so darn expensive? You know, $250,000 a year, $500,000 for some drugs, not in our space, but it's over $1 million a year. So why does it cost so much? So we heard about the development process earlier, and you know, the development costs for treating patients, whether it's 10 million patients or 100 patients, it's the same cost. You still need to do all of those steps, okay? Clinical trials can actually be much more difficult to do with rare diseases because it's hard to find the patients. We know how hard it is to get the diagnosis. If people can't get their genetic tests, they can't get their genetic counseling. People don't know where, what they're even diagnosed with. And often clinical trials are done in, you know, centralized urban centers, and if patients are 7 or 8 hours away, you know, they don't necessarily want to be part of a clinical trial because it's just they'd have to constantly be coming back and forth for this. So that can lead to increased costs. Sometimes patients have to fly down to the US because there's only sites in the US. These are all costs that accumulate. And in some countries, there may be less than 100 patients who even have the disease that is eligible, you know, that could be eligible for treatment. And you have to put all this infrastructure in place. You have to import the drug. You have to have your warehouse. You have to have your patient support program. So all of these things cost, and that cost has to be defrayed by so few patients, because this isn't Ozempic where you're treating 10 million patients. You know, you might be treating 100 patients. So that's why rare disease drugs are so expensive. So I just wanted to touch on that. And so in summary, it's a complex world to get a drug approved in Canada and to get it reimbursed. Lots of different government agencies. But those government agencies have a really important job to make sure that we bring safe, effective drugs into the country and that we're bringing value to the healthcare system. This can take 3 years, and that's why, as Canadians, we've learned to be patient. We do have multiple payers in Canada, so we have private, we have public, sometimes people have both, but it's a lot for patients to have to navigate, and that's really where patient support programs can really help you out, can help physicians, can help patients, can help caregivers. Many manufacturers actually help accelerate this to get access for patients sooner so that they're not sitting there waiting and being frustrated by that. And the National Rare Disease Strategy, it's designed to help, but it's been a bit of a slow process. So obviously the patient voice is important there. There's going to be the Fall Cord Conference in Montreal at the end of November. So I know Durhane will be working hard on that. So anyways, that's it. If there are any questions for myself or Dr. Chakrabartty, I'm not sure, Alon, how much time we have, but he's got...
We can take a few questions.
He's got microphones, so...
Two fascinating presentations. I think we could probably spend all morning talking about both of them, but we do have a few minutes for questions. Dr. Chakrabartty, why don't you come back up here if you don't mind, just in case there's any questions for you, and maybe we could bring a microphone to the podium, yeah, or to the table, and then just see if there's any questions.
Alon, I think there's someone over on this side.
Ah. You're in my blind spot.
Yeah, my question was with regards to the depleters. Now, I understand that these are to clear the deposits from the heart. Now, is this strictly an ATTR, or does it work with AL patients as well?
Sure. It's on? Okay, thanks. Yes, that's the common theme for all of the amyloid diseases. Of which there's like over 30 of them, and and so that's the common theme that runs through this whole family.
And sorry, can I just add to that? That was talking about the depleters in the heart, the fibrils. Is that also for the kidney, or is there no research on that?
Yeah. Sorry. So let me see if I understand this. You want to know whether there's whether there's a unique deposition that occurs that's organ specific? Is that that's definitely true. I mean, every amyloid disease has its own organ specificity, and they vary. The common so some places are more, I mean, so there's a variation, and it also varies with the amyloid type. So that's it's a variety. So
if I could expand on that question, Dr. Chakrabartty, over here, yeah.
The question I'm having trouble with. Yeah.
So. I can understand, the fibrils, the pancake stacks. I love the way you explain these things. I always learn something when I'm talking to you. I understand how there's kind of similar conformation for different amyloid precursor proteins to those fibrils. But the binding to the TTR monomers, that part I have a harder time wrapping my head around, because those binding sites would presumably be different than kind of AL precursor protein binding sites, the light chains. And so that part would differ between the two diseases, binding to the kind of the pre-fibril stage? And also, if the monomers are unstable, how does an antibody bind to it? Does that matter? So 2 questions, I guess.
Yeah, okay. So I'll take the monomer question first, because it's easy to answer. There's usually a small amount of monomer always present, and so because there's that there, so the antibodies bind to that monomer pool, and then when new monomers emerge, the antibodies will be there to mop it up. So that's the process. Also, so the amyloids that is deposited in your nerves is different structurally from the amyloid deposited in your heart. So the amyloid in our heart, we call that type A amyloid. and the ones in the nerves are type B amyloid. So there's a structural difference between the two. So we think what happens is you get the layering of the pancakes, and then after you get the layering of the pancakes, other proteases and things come along and nibble on it, and that changes it. And then one of them becomes type A, and the other one becomes type B amyloid. So, yeah.
Fascinating.
Okay, so so that level of detail we do know about cardiac and nerves. We don't know yet. Yeah, somebody has to do those studies. Yeah, thanks.
I have two. First one is about the silencers. So if we take CRISPR-Cas9 out of it and just focus on the other silencers, in your slide you mentioned slows progression. I'm just wondering why isn't it stops progression? Can you explain that? And then the second question on the drug trials. This is more of a knowledge piece one, but how are we ensuring that our specialists in each of the regions have clear line of sight on all active trials in Canada that are going on, and also the ones that are going on in the United States that would be an option?
I could try to take the second one.
Okay.
Yeah.
Okay. Right. Yeah, the first one. Can you give me a?
So it was why doesn't it completely stop progression in the silencers?
Okay. Right.
Yeah.
Right. Yeah. So that's one difference between the CRISPR-Cas9 and the RNA silencers, the RNAi and the oligo antisense oligos. So with CRISPR-Cas9, I think I don't remember the actual percentage, but it's really high, the percent reduction. So that drops it a lot. But with the other ones, there's still you don't go down to zero. Yeah, you go down to like 30% or so. So there's for the RNAi and the antisense oligos, that seems to be a plateau region.
Yeah, my understanding is kind of between 80 and 90%. Actually, for the silencers and even the CRISPR-Cas9 is a drop in reduction. So I don't think you go to zero for either category of drug. But we're still learning about this, especially for the CRISPR. Right, it's still very early days, and all we have is the phase 1 data. And then the second is, I think you probably know what I'll say to this, is that access to clinical trials and awareness of clinical trials, I think, will also be a priority for the Canadian Amyloidosis Society, and kind of go hand in hand with kind of a care map, right? So where is care available and which centers are doing clinical trials, both for AL and ATTR, to make that information more readily available both to healthcare providers and to patients. So kind of a common source to just look up, okay, where are these clinical trials going on, and who can I contact if I'm interested. I think that's a very important priority as well.
Yeah, a question related to your a question related to your earlier answer on the different type of protein deposition, depending on the deposition site, so nerve versus heart. Do we know if that has an impact on the availability of the binding sites for the antibodies? Does that mask the in one organ versus the other?
Yeah, so only sort of qualitative experiments have been done to address this. But one particular antibody, you can see how it stains the amyloid in nerve tissue, kidney, and so you can compare that sort of qualitatively, and so the antibodies there isn't one particular type of amyloid that I know of so far that the antibody doesn't recognize. So yeah.
Great, thanks. Well, I think we could spend the rest of the day asking questions to you both, but we should move on. So thanks again to Dr. Chakrabartty and to Colleen Coxson.